This talk highlights our recent work which investigates the mechanisms of survival of chronic lymphocytic leukemia cells in the lymph node microenvironment. So in the current era of targeted therapies, there has been a very strong, there have been significant advances in therapy of CLL with inhibitors of B-cell receptor signaling. However, those B-cell receptor inhibitors do not provide for complete responses or disease eradication. And we believe that part of the reason why is because in addition to B-cell receptor signaling pathway, there are several other pathways in the lymph node which are very important in promoting malignant B-cell survival. And so one of those pathways is actually BAFF, B-cell activation, B-cell activating factor. And so BAFF is ubiquitously expressed as a ligand by many types of cells in the lymph node. And so how do you study it? So what we actually have done is we mimicked the microenvironment BAFF expressing niche by using CHO cells, so the sort of which we use the stromal cells and co-cultured them with CLL cells. And so those cells expressed BAFF and continuously delivered BAFF to the neoplastic primary B cells, which we took from patients. And so what we wanted to study is to figure out how BAFF signaling behaves in a setting of B-cell receptor signaling inhibition. And what we found is that BAFF actually induced B-cell receptor signaling by itself, just like IgM cross-linking, what you would do in CLL cells in the lab, normally in vitro to induce B-cell receptor signaling. BAFF ligand just did exactly the same. It induced activation of B-cell receptor-associated kinases in CLL cells, including BTK, ERK kinase, and SYK kinase, as well as some others that we looked at. And so what we determined further is that obviously it resulted in activation of NFκB pathways, both canonical and non-canonical, and the canonical pathway activation happened via this BAFF-BCR crosstalk and was abrogated by B-cell receptor signaling inhibitors. One other piece of this work is that it seemed that in this BAFF expressing conditions, as an important anti-apoptotic element was induced called MCL1. And it's been shown in the past that MCL1 predicts resistance to conventional chemotherapies in CLL and also to some novel agents like BCL2 antagonist venetoclax. And so we have shown that one of those BCR signaling inhibitors called entospletinib, which targets SYK, spleen tyrosine kinase specifically, but not other BCR inhibitors, not ibrutinib, not idelalisib, actually abrogates this BAFF-mediated induction of MCL1. So we believe that this is a very interesting point there where entospletinib targeting SYK specifically disrupts this cooperation between BAFF and B-cell receptor signaling and thereby counters BAFF-mediated CLL cell survival. So in part, this work is actually leading to a clinical trial that we are launching together with Gilead, a phase 1 clinical trial of entospletinib in combination with the brentuximab, which we will be opening in the United States very soon. So this is basically direct bench-to-bedside translational work.
Okay. And what will be the endpoints for that trial?
So the phase 1 component is looking at safety for this drug combination, and phase 2 is specifically focused on CLL and is looking at efficacy. How much response are we achieving with this combination?
Are there any plans to go back to, say, for example, an organoid model to figure out some of the interactions that you said there, and any more, for example, pharmacokinetic states?
Right, so we certainly will have a lot of translational pharmacodynamics endpoints on the study, and so we will be looking at some of the PD endpoints that we have identified in the lab, looking at MCL1 expression and NFκB activation and how it will be affected by treatment with entospletinib. And generally, this work opens a whole can of worms in terms of how exactly BAFF induces B-cell receptor signaling. There are several receptors there which can respond to BAFF, and we don't know exactly how this interaction occurs. The other thing is there is another ligand called April, which is sort of BAFF ligand cousin, and we also don't know whether it's also very prominent in CLL microenvironment, so we don't know if that might also be important, whether that would also induce BCI signaling just like BAFF does. So it's certainly, there are certainly a lot of unanswered questions there.
Okay. And on a final note about SYK, we heard from, I think it was Dr. Hamlin yesterday, talking about cerdulatinib, a dual JAK-SYK inhibitor. Do you think there might be some crossover there?
Possibly. So, yeah, so the entospletinib is a very selective SYK inhibitor. So there is some history there with fostamatinib, which was a fairly dirty kinase inhibitor, so it inhibited a bunch of things in addition to SYK. So entospletinib is a better designed drug, which is fairly selective inhibitor of SYK. In terms of concurrent JAK-SYK inhibition, I think it's a very good idea. I think time will tell in terms of what you really need to achieve, how much inhibition you would need to achieve. I think we would need to have good PD data, whether we are really saturating the target PD-wise in terms of inhibiting all the downstream signaling pathways appropriately. Entospletinib has not been associated with a whole lot of toxicities. A phase 2 study in CLL single agent has shown good tolerability of the drug. With JAK-SYK inhibition, I guess it remains to be seen how well that will be tolerated, because you are certainly adding another very ubiquitous target. But yes, that certainly there certainly could be a cross-dissemination there as well.